Hello everyone, my name is Kimleo Alcuetas.
Hello everyone, I'm Shinomi Camero Altake.
Hi everyone, I am Ken Taron A. Alminario.
And we are from BSN Science 2A.
Good day everyone. Our topic is all about Introduction to Biology.
And the learning outcomes are give practical application to scientific method, determine the domains of life, identify the different cell structures and cell types, and discuss the methods used for studying cells.
Scientific method, mathematical and experimental technique employed in sciences. More specifically, it is the technique used in the construction of testing of scientific hypotheses we usually used in researches. Now that we learned on scientific method, our next topic is all about the three domains of life. So what are those? Number 1 is the archaea. Number 2 is the eubacteria. And number 3 is the eukarya. But before that, we have to learn first on what domain is. In biology, a domain refers to a largest of all group. There are only 3 groups of this level: the archaea domain, bacteria domain, and the eukarya domain. Each domain contains collection of organisms with similar properties and evolutionary histories and scientists that organize them.
Any of the groups of single-celled prokaryotic organisms that have distinct molecular characteristics separating them from bacteria are called archaea.
Number 2, eubacteria, also known as true bacteria, are single-celled prokaryotic organisms that have a range of characteristics and are found in various conditions through parts of the world.
Number 3 is eukarya. Eukaryotes are organisms whose bodies are made up of eukaryotic cells such as protists, fungi, plants, and animals. Eukaryotic cells are cells that contains nucleus and organelles and are enclosed by a plasma membrane. So next is we will define what is a cell. A cell is the building blocks of all living things. Example is the human body. Our body is consist of trillions of cells that provide structure to ours. They take nutrients from our food and convert it into energy.
And we have the parts of the cell. First is the intermediate filament, rough endoplasmic reticulum, nucleus, nucleolus, chromatin, Golgi apparatus, Golgi vesicle, cytoplasm, mitochondrion, plasma membrane, microtubule, centrosome, microfilament, lysosome, smooth endoplasmic reticulum, secretory vesicle, peroxisome, and vacuole.
After naming the parts of the cell, we will now define a few of them. Cytoplasm. Cytoplasm is made up of jelly-like fluid called the cytosol and other structures that surround the nucleus. Cytoskeleton. The cytoskeleton is a network of long fibers that make up the cell's structural framework. The cytoskeleton has several critical functions, including determining cell shape, participating in cell division, and allowing cells to move. Endoplasmic reticulum. This organelle helps process molecules created by the cell. The endoplasmic reticulum also transports these molecules to their specific destinations, either inside or outside of the cell. Golgi apparatus. The Golgi apparatus packages molecules processed by the endoplasmic reticulum to be transported out of the cell. Lysosomes and peroxisomes. These organelles are the recycling center of the cell. They digest foreign bacteria that invade the cell, rid of the cell of toxic substances, and recycle worn-out cell components. Mitochondria are complex organelles that convert energy from food into a form that the cell can use. They have their own genetic material separate from the DNA in the nucleus and can make copies of themselves. Nucleus. The nucleus serves as the cell command center, sending directions to the cell to grow, mature, divide, or die. It also houses DNA the cell's hereditary material. The nucleus is surrounded by the membrane called the nuclear envelope, which protects the DNA and separates the nucleus from the rest of the cell. Plasma membrane. The plasma membrane is the outer lining of the cell. It separates the cell from its environment and allows materials to enter and leave the cell. Ribosomes. Ribosomes are organelles that process the cell's genetic instructions to create proteins. These organelles can float freely in the cytoplasm or be connected to the endoplasmic reticulum.
Since the cells are the smallest unit of life, most cells are so small that cannot be viewed by our naked eye. Therefore, scientists must use microscopes to study cells.
Since there are many types of microscope that we use today, we will name few of them. Number one is the most common microscope that we use, simple microscope, which invented on 17th century by Anthony van Leeuwenhoek. While the others are the following: compound microscopes, stereo microscopes, contact microscopes, scanning electron microscopes, and transmitting electron microscopes are mostly invented because of innovation.
But Zacharias Johnson, credited with inventing microscopes for millennia. The smallest thing humans could see was about as wide as human hair. With the microscopes invented around 1590, we saw a new world of living things in our water, in our food, and under our nose.